Well, good morning, good afternoon, and good evening, folks. Before we start, we wanted to say thank you for your participation today. Whether your institution allows 25, 50, or 100% capacity in lab, we appreciate your time away from it. As you'll see here on screen, this is our agenda for the day. We've got a few hours of really informative speakers, starting with a couple of talks by your sponsors today, Arima Genomics and Agilent Technologies, and then 3 interesting talks from the Asia-Pacific region. Really exciting stuff. We've had over 100 registrants for today's event, and this is going to be an incredibly fun and informative few hours of insights during our inaugural APAC co-webinar with our friends at Agilent. My name is Christian Nelson. G'day, good to see you. I'm the Global Sales Director at Arima Genomics, and I'll be your host and MC for today's webinar, Epigenetics in Human Health and Disease. Let's see, we have a few slides we wanted to go through just to talk about a little bit of housekeeping notes. I'm sure it's not your first webinar of 2020. There's a few tools out there, and we're using WebEx today. There's a couple of functions within WebEx, the first being the chat. This is not the place to put your questions for Q&A sessions later. This is the place if you have technical assistance just to chat us, and we'll be happy to try and take care of what we can on our side. The third in the middle here is the polling box. So towards the end of the day today, we'll have a few questions for you just from a polling perspective to try to gain your insights on how you thought the day went and what you thought of the content. So stay tuned for that. And then finally, there'll be a box on the right, and this is a great place for you to put any of your questions that you have at any time, and we'll try to address those again at each individual end of the section from that speaker, and if not, we'll save any questions for the very end. We have about 15 to 20 minutes parked for questions and answers. Okay. So, and we have a number of registrants, and we appreciate all of your time. Just wanted to get to it here and start the agenda. I'll be introducing all the speakers, and while they're preparing their screens, just sharing with you a little bit about their background and their bio, and off we go. Perfect. So first up, I think we have the screen coming up, is Arima Genomics' very own Senior VP of Science, Dr. Anthony Schmidt. Anthony means a lot to Arima. He's the foundational scientist behind the extremely impactful Arima HiChIP platform. He's the lead developer of HiChIP and Capture-HiC solutions, and I wish I could share with you what products we have up our sleeves for you for 2021, but you'll have to stay tuned. He's well-trained, well-published in the field, and a generally good person to work with. Anthony, the floor is yours.
All right. Thanks, Christian.
Good to see you.
Yeah, thank you. All right. Cool. Well, thanks for the introduction. I'm thrilled to be here talking to everybody virtually. We haven't met in person or virtually before. I'm Anthony, lead the science at Arima. I've been with the company since before it was a company, so since its inception, and I'm excited to speak with you all today. Okay. A little bit about Arima, just in case you haven't heard much about us. We are a biotechnology company aiming to accelerate the understanding of the genome sequence and structure information. We fundamentally believe that the structure is as important as the sequence in understanding human health, disease, and species evolution. And the way that we try to help fill these gaps in knowledge is by facilitating access to high-quality sequence and structure information in the form of reagent kits as well as services. So our first product here, the Arima HiChIP Plus kit, we launched that back in 2018. The original use was, characterizing, genome folding at a genome-wide scale via HiChIP. And then since then, over the past couple of years, we've launched 2 different, targeted versions. One called HiChIP that I'll talk about today, and another called Capture-HiC that I'll also talk about today. And this type of 3D DNA folding information is also really important for a whole other sort of area of research called genome assembly. We also have some products in that space, and we're really just interested in helping unravel the genetics of both humans as well as other non-model organisms. So today I'll talk about a few different things. I'll start with Arima HiChIP. I'll spend most of the time talking about Capture-HiC, one of those targeted versions of HiChIP, and then I'll finish with just a little bit about HiChIP. And hopefully this sort of provides an introduction to some of the tools that we offer, but that will also sort of serve as an introduction to what we'll hear about in the talks that will follow. So first, really just to set the stage, a little bit of motivation and background on the 3D genome. This is from a review from my PhD mentor, Bing Ren, back in 2014. All it's really showing here is that the genome is organized into hierarchical layers of genome organization. So really looking down into an interphase nucleus, you might see chromosomes occupying their own distinct territories within the nucleus, which is shown here by the chromosomes in different colors. This has been supported by microscopy data, and some of really the initial HiChIP data sets realized this discovery through really an abundance of interactions within chromosomes and a dearth of interactions between chromosomes, supporting this idea of chromosome territories. As you zoom in, sort of on the left, hopefully you can see my cursor. Other structural features like chromosomes tending to organize sort of next to the nuclear lamina and other parts extending more towards the nuclear interior. And as you zoom in a little bit further, let's say on an individual chromosome, you see these self-interacting domains of chromatin, also called TADs, shown here, kind of on the megabase scale. And these TADs are demarcated by architectural proteins like CTCF that sort of serve at the boundaries of those domains. And really the concept there is you can have two different loci, let's say point A and B, that might be really close together along the linear genome. And, if the folding of the DNA was random, you might expect some type of, crumpled up ball of yarn where A and B are also close in three-dimensional space. But in fact, the folding is not random. You can have two pieces, A and B, that are close together along the linear genome, but actually spatially segregated into two, neighboring TADs in different regulatory environments. And back over here to the left, as you zoom in further, it's kind of on the scale of tens of kb to hundreds of kb, these individual chromatin loops that are being depicted here that are also held together by architectural proteins. And in some instances, these sort of interactions that are happening here right at the base of the loops can bring together cis-regulatory elements, promoters, enhancers, and what have you. And what that sort of looks like when you look at HiChIP data is you see these triangles along the diagonal. And if you zoom into one of them, these sort of very fine point-to-point interactions of these dots off the diagonal really represent that point-to-point interaction that's happening at the base of those loops. And just to give an example of why this is important, really these tools have been used for decades now, and this is an example from one of our customers over at NYU here in the U.S., the Iofantis and Syragos labs, who used HiChIP to study the three-dimensional landscape and regulatory landscape of leukemia. And what we're looking at here first is really a zoom in of a HiChIP contact matrix right around the MYC locus. And this is in healthy primary T cells that were isolated subjects. What you can see here is a gene is kind of nestled in right into this topological domain, and then downstream of that, approximately 2 megabases away, is a series of cancers that in this configuration in healthy T cells can't access MYC because it's in its own sort of three-dimensional domain here. But really surprisingly, in leukemia, there's a strikingly different, three-dimensional conformation around the MYC gene. There's almost a coalescence of these two, domains that sort of fuse into one separate domain. When you, zoom in and you look at the underlying, transcriptome or chromatin landscape, what you see here is that in the healthy T cells, when you look at CTCF binding, you see, of course, CTCF at the edges of the two different boundaries here. And in the healthy T cells, there's actually a CTCF peak sort of sitting right there in the middle, which might explain the demarcation of those two domains. And then that CTCF binding site goes away in the leukemia cells, which may explain sort of that change in three-dimensional folding that you see. And also what happens in the leukemia cells, if you look at the K27 acetylation ChIP-seq data down here, is that signal around the super-enhancer regions turns on in leukemia. So to sort of simplify the concept, really with sort of the old, linear genome paradigm, you might have MYC over here, what will be an active enhancer about 2 million base pairs away in a healthy individual. When that oncogenic transformation occurs, you observe more MYC expression. Maybe you see, I think in this case, CDK7 binding at the enhancer. And then if you treat with, let's say, a drug that is an inhibitor for CDK7, you observe the expression turn off. But there's really not a lot of insight into how this process is happening. And with the 3D genome paradigm, thanks to this paper and others, in the healthy state, MYC and the enhancer are in two separate spatial domains that I'm showing here. And then in the oncogenic state, those two domains merge together. Now there's that spatial interaction between MYC and the enhancer. And then when you treat with drug, they show that you see a reduction in the interaction between MYC and that enhancer region. It really provides more insight into how these types of gene regulation mechanisms are occurring. So that being said, I'm just going to give a little bit of information about our Arima HiChIP workflow. We start with fixed cells over here on the left. We digest with a restriction enzyme cocktail, essentially, cutting up the DNA into smaller pieces. And, one of the important things here is that when you're looking down on this cell, the gray piece of DNA and the blue piece of DNA back here are being held together in 3D space, but they're actually, linearly very far apart along the linear genome, but they're being held together in space. And you fix that with formaldehyde, and there's really just three very simple steps. Digest with restriction enzymes, fill in the digested ends with a biotinylated nucleotide that you can see here in the little red dot, and then you ligate them back together. So what was two pieces of DNA that were not adjoined, but they were held together in 3D space, are now physically linked together by that ligation event. And then you purify away the proteins, and you have your purified DNA. This whole process takes about 6 hours using our kit. This is sort of what we refer to as the core high-speed technology. And then we have a quality control checkpoint here after this is over. So really we have very flexible sample input requirements, tissues, blood, cell lines, what have you. This whole thing happens in 8 steps. It's a single tube chemistry, and by that I just mean from starting the reaction with your lysis buffer, you just keep piling on reagents all the way through the very end of the workflow. There's no spins, there's no fine-tuning digestion conditions or anything like that. It's pretty plug and play. The whole thing takes about 6 hours, and like I said, we have a quality control checkpoint right at the end there. And really to follow that, we just build Illumina sequencing libraries with a slightly modified protocol. You can take advantage of the Kappa Hyper prep reagents or other library prep reagents that you might have in your lab. We have optimized protocols for various different commercial library prep kits. One of the things that always comes up here is sort of how low can you go in terms of sample input requirements. Really, we say about 50,000 cells or even less is kind of the bottom of what we recommend for genome-wide HiChIP analysis. There's this one recent publication from the Akbarian lab at Mount Sinai here in the U.S. That has analyzed and published our kit down to about 6,000 cells from postmortem brains. And we offer this as a kit or a service format. You can read about the various different testimonials on our website. I think the technology speaks for itself. It's been around now for a couple of years, and we've been very happy with those who have been using it. There's about 30 publications now that have described the use of the kit across various different domains of research. And we've been so pleased with this core technology and its utility in different forms of research. But over the years, we've also heard a lot of feedback that genome-wide analysis is sort of not the appropriate way to best address many different biological hypotheses that are more targeted in nature. Like if you're really interested in, let's say, CRISPR perturbations of a particular locus, and you want to measure just the conformations that might be changing around that loci, it doesn't make sense to sequence an entire genome-wide HiChIP map. But the two different targeted technologies for that are called HiChIP and Capture-HiC. I'm going to talk about Capture-HiC first. So, I think it's nice to explain Capture-HiC relative to something that most people have already heard of or maybe have, experience with, such as exome sequencing. Exome sequencing has been around for a long time, and the workflow is you extract your DNA. Sometimes you have to shear your DNA into smaller pieces. you add your sequencing adapters and you PCR amplify. But before you would traditionally sequence, you introduce capture sequencing probes that hybridize to the exonic sequences in the genome, or whichever sequences you want the probes to hybridize to. You enrich those molecules and then you sequence just the ones that have been enriched, and that's your typical targeted sequencing workflow. And with Capture-HiC, it's the exact same concept, but instead of building just a traditional genomic DNA library, you build a HiC library instead. So it's all the same steps that you would do using our kit to make your genome-wide HiC library. You go through all the same steps, and then right here, after you amplify instead of sequencing, then you add your biotinylated probes. We've really had the pleasure of partnering with Agilent for about the past year or so, building this integrated, optimized protocol. We have a application note that's available, I believe, on the Agilent website. You can also share it with you that describes all the data I'm going to be talking about in a second. So we add the Agilent probes, the Agilent hybrid reagents, then we PCR amplify and then we sequence, and then that final product that comes out of this is the Capture-HiC library. Really, I think to get all of us thinking about how this could be a useful tool in your own research, there's sort of different flavors that we think about when it comes to Capture-HiC, and I've tried to illustrate a few of them here. The first one, which I'll show more data for, is the concept of region Capture-HiC, where you have a gene of interest, and maybe you want to tile capture probes to enrich for that gene, as well as the local neighborhood around that gene to capture 3D regulatory events that are happening right around that gene of interest. So you might do something like this. This is a paper that took a similar approach. It's not an Arima paper, but it's also just a nice reference point to see the application of region Capture-HiC. A second application is really geared at helping interpret the function of non-coding GWAS hits, with the idea being that perhaps a non-coding GWAS variant is somehow affecting a target gene through DNA looping. And if you design probes to enrich around these GWAS regions, you will then discover which genes might be implicated in that disease. And then lastly, the idea of designing probes right around the promoter regions of all of the promoters in the genome or a subset of promoters in the genome that may be of interest to your research. And the way that we partner with people on this is really we help along the entire way. If you send us a list of genes that you're interested in or a list of regions, our team will help with the probe design. We use the Agilent Sure Design software suite for probe design. We have stock promoter Capture-HiC panels for mouse and human, and we also do a lot of different custom designs based on your own needs. Through this design, we then iterate, so we can share the design files with you. You can review all of the designs, all the regions that are being targeted. We can iterate as needed. And then once we're both happy with the design, we link you up with Agilent to order the probes, and then effectively the Arima HiC kit plus the Agilent probes is the combined set of reagents for the Capture-HiC workflow. And I just wanted to show a bit of internal data that's also found in the application note. One of the experiments that we did really to validate this workflow is we designed probes around a series of oncogenes and tumor suppressors or other interesting gene regions listed here. And what I mean by BCR 620 kb is if the BCR gene, let's say, is living in this topological domain, I just put probes around the BCR gene, and then we can leave everything else, behind. We can do this for about 10 different regions in the genome in this example. And we did this on 3 different non-cancer cell lines and 7 different cancer cell lines. And of course, I also picked these because several of them are ENCODE cell lines, so we were able to pull together lots of other public data and build a story around integrating Capture-HiC with other omics data sets. This is a busy slide. I'm just going to block up some of the things that are less useful here. If I can go back. Here it is. So in terms of sequencing depth requirements, one of the things I'm going to be talking about is how the sequencing requirements are substantially less than genome-wide HiC. Of course, that makes sense. But just to put some numbers around that, we're sequencing about 90 million read pairs per sample. And in terms of the capture efficiency, about 60% of all of the read pairs are on target, and that equates to about 150x fold enrichment. So what does this data look like? What you're looking at the top here is a genome-wide interaction matrix where all the different chromosomes are along the x-axis, all the different chromosomes are along the y-axis. Really, at this sort of zoomed-out view, the data doesn't really mean anything, but what I wanted to point out is that with the Capture-HiC data, you can zoom in to all these very small regions that you captured and pull out these beautiful, targeted, high-resolution HiC maps. And what we're looking at here happens to be the region around the EGFR gene. So you see the HiC map here. This is visualized in an open source program called JuiceBox, developed by the Aiden Lab at Baylor. And what you're looking at here is not only the Capture-HiC data, but then CTCF ChIP-seq, K27 acetylation ChIP-seq, and all these other omics data sets that you can visually integrate into one view using JuiceBox. You can do that for all the different captured regions from the panel, P53, GND, P10, NTRK, and all of the data from this experiment is openly available through our FTP. You just have to ask for it if you want to have a look. I wanted to really step through what I think is really the main advantage of Capture-HiC, and that is the ability to push the resolution of the analysis to really unprecedented levels when it comes to HiC, and that is sub-kilobase resolution. And to take you through that story, I wanted to show an example in K562 leukemia cells around the BCR gene. And really to make this point, I want to first bin the data at fairly low resolution. So this is what the Capture-HiC data around BCR looks like if I were to bin the data at 25 kb bin size. And what you can tell from this sort of coarse view is that there appears to be some interaction happening sort of off the diagonal in this space, and it appears to be involving this bin and these two bins down here. When you take those bins and you trace them up to what's happening along the linear genome in those regions, well, you can see that there's the BCR promoter, there's a couple different CTCF and cohesin co-bound peaks. This appears to be an active promoter based on a K27 acetyl signal. And then over here, again, there's two different CTCF cohesin co-bound peaks. You can't really say with much precision whether the interaction is involving the promoter or if it's just upstream of the promoter. Right. Now I can take the same data and just change the bin size, and it has the resolution to support that, and this is sort of what it would look like. Again, you can tell that there's some type of looping interaction that appears to be happening, but again, when you trace that back along the linear genome and you ask, what's there along the linear genome, again, you can't really resolve whether this loop is anchored at the promoter, and if that's important to you, you don't have the resolution or the precision to gain that insight. But with Capture-HiC, from just about 100 million reads or a little less, you can take the same data and actually bin it at 500 base pair resolution, which is what you're seeing here on the right. And there you can tell that this, looping interaction really involves the promoter of BCR and this CTCF cohesin co-bound peak downstream of it. Right. So this analysis that you're seeing here would have taken about 8.3 million reads to obtain this type of resolution if you were just sequencing this 600 kb region around BCR. So lastly, time, a few minutes, I wanted to talk about HiChIP. Really, HiChIP was published back in 2016 for the first time by Howard Chang's lab at Stanford. Shortly thereafter, we had a paper from Bing's lab describing something very similar. It was called PlaqueSeq. It was one of the last things that I had the opportunity to work on when I was in Bing's lab. So it's been nice to get that hands-on experience and then apply that to Arima. And similar to how I introduced Capture-HiC, HiChIP is in many ways very similar to ChIP-seq, where ChIP-seq, of course, is sharing your chromatin, adding your antibody, doing your IP reaction, adding your sequencing adapters, and amplifying sequencing. And with HiChIP, really just that proximity ligation-based workflow where you capture the DNA-DNA interactions within intact cells in situ happens first. So same sort of HiC chemistry digestion, fill-in, and ligation. And then these proximally ligated chromatin really become the substrate to the ChIP reaction, where you add your antibody, you do your IP, and then you build your sequencing library. So at the molecular level, the way that it's different is the molecules that are getting enriched have that sequence content right around the immunoprecipitated protein, just like ChIP-seq, but these molecules also carry that three-dimensional interaction that's associated with the immunoprecipitated protein. Right, so this technology inherently captures the 1D protein localization information like ChIP-seq, and then of course all of those interactions that are associated with that protein. So if you enrich for something like K4 trimethylation, you can characterize those interactions associated with active promoters. We have optimized protocols for the diagenode shearing platforms as well as Covaris. I want to say that we've had a wonderful collaboration with Covaris over the past year, and to direct you to our poster we did with Covaris at AGBT 2020 this past year. And then we offer this as a kit or service format. I want to just briefly touch on this nice collaboration we've had with Ming Hu's lab at the Cleveland Clinic. Ming developed some of the first HiChIP data analysis tools, and we've really built on top of that. We've tried to build more user-friendly data reporting features, like by default, once the HiChIP data has been run through this pipeline and loops have been called with statistical significance, we also look at the chip enrichment of the data. We automatically output files that can just be dropped right into the WashU genome browser, so you can visualize your loop calls next to your other omics data sets, and a quality control table to look at various different layers of QC. So what do these data look like? This is a snapshot of the MIC locus. Really, I want to draw your attention to the bottom two tracks here. This is K27 acetylation ChIP-seq data around the MIC locus. This is from ENCODE. And if you plot the HiChIP data just like you would ChIP-seq data, the sort of 1D signal track very much mimics ChIP-seq, saying that the data is equally as enriched as ChIP-seq. But of course, ChIP-seq doesn't give you all the three-dimensional connectivity information like HiChIP does. So if I now add on the loops that are called from the HiChIP data, it paints a different story of lots of long-range gene regulatory interactions that are called by the MAPS program. Overall, we see about 75 to 85% of the loops that are called in one replicate are also found in the second replicate. So to wrap up here, we've had the HiChIP kit out on the market now since 2019. We've had several researchers successful with the kit. You can read more about this on our website. I just wanted to do a quick plug. We are announcing new capabilities of our HiChIP kit next week, and I'm going to be talking about a new protocol that's been validated for transcription factors, frozen tissue samples, improved bioinformatics capabilities to call ChIP peaks from the HiChIP data, as well as improved usability features. And you can contact us or check out our website for the registration information for that webinar. I want to thank our advisors, Yob, Jesse, and my former mentor, Bing Ren, as well as our funding support. Thank you.
Okay, thanks, Anthony. We really appreciate the time and the content. We have a question here. I'll jump right into it. We just have a minute or two. Regarding Capture-HiC, where are the capture probes targeted within the BCR? Why is there still a significant amount of reads along the linear?
The probes were targeted, really tiled across the entire locus. Really, unless that there was some sort of repetitive content, I believe that would be masked by the Sure design program that does the probe design. The probe should be more or less tiled across the entire locus that we defined.
Okay, perfect. Let's see, one more. Thank you, Anthony, from joining down in Australia. Do you plan to update the protocol for using the Arima HiC kit specifically with FFPE samples?
I'm sorry, who was that question from?
It was Joe down in Australia.
We do not have an FFPE protocol right now, but I think what we do have is we there's a preprint that's available now that describes our first FFPE HiC data. I think if you follow my Twitter, you might see it shared. So you can check out the details there, as well as some of the data in that figure is now a preprint.
Who doesn't follow your Twitter? Who doesn't? I think that's all the time we have today. Really appreciate it, Anthony. Thanks again, mate. Cheers.
Cool, thanks.
Okay, we'll keep things moving here. And just a little reminder for everyone, on the right side of your screen is the Q&A box, so thanks for your questions so far. We've had a couple of others that we'll share here towards the end. Wanted to move on to the second talk in the morning from Dr. Chiang Li, who is a Senior Field Application Specialist at Agilent. Chiang has been with Agilent for over 10 years. I'm sure he can address many, if not all, of your Capture-HiC related workflow questions. Prior to Agilent, Dr. Li gained his Ph.D. from Curtin University while characterizing the MHC Class II region in sheep. Chiang, thank you very much and welcome.
Well, thank you. Thanks, Christian. Good morning, everyone, and thank you for joining us today at this Arima Genomics and Agilent webinar event. So as Christian earlier introduced, my name is Chi, and I am the Agilent Field Application Scientist for the Diagnostics and Genomics group here in the SAP region. So what I'd like to do in the next 20 minutes or so is to just provide a very brief snapshot of the Agilent NGS portfolio, which not only includes solutions for targeted sequencing, but also for other epigenetic-related applications of interest. Now, as I'm sure many of the audience here today are very familiar, next-generation sequencing, or NGS, is a technology that has enabled the research community to better understand the genetic makeup of the species of interest. And while NGS provided an immense amount of data, it is not without its challenges, be it the cost of sequencing or handling that large amount of data that has been generated. And more importantly, there is the age-old question of whether should we be generating data on everything, regardless if it is of interest or not, or should we be focusing only on those regions that we are interested in and targeting those regions that are related or relevant to our research. And it is through this debate that the concept of targeted NGS was explored. Now, the concept of a method to enrich for the DNA by hybridization with tagged oligos gave rise to the Agilent target enrichment solution that is SureSelect. The SureSelect technology utilizes 120-mer cRNA-based cRNA biotin labeled bait or probes to capture the DNA from the region of interest. Now, Anthony showed very elegantly in that diagram earlier on in his presentation. So these cRNA baits can be designed to any regions of interest, and hence it is easily customizable. And that whole enrichment workflow or process with SureSelect is done in solution. And therefore, the samples can be easily multiplexed, and the process can be automated. And from the first development, the SureSelect portfolio has expanded to cover the whole targeted NGS workflow, barring the sequencer in the middle. This includes library preparation solution that can accommodate a range of sample types or input amount while maintaining high levels of library conversion efficiency. Then, of course, there is the core of the enrichment workflow, where the target enrichment probes or baits can either be catalog designs, such as a whole exome, so targeting all the coding exons, or a custom approach, whereby the user, such as yourself, can customize a panel specific to your requirements. The NGS workflow also requires sample QC in order to ensure quality control of the starting material, such as integrity of the starting input genomic DNA, total RNA, and even cell-free DNA. Or for the sample QC of the process sequencing library, so the library that you've created prior to sequencing, you'll also be doing sample QC on them. These can be checked with platform solutions such as the TapeStation, bioanalyzer, as well as the fragment analyzer. In addition, there is the automated solution to facilitate not only an increase of sample throughput, but also for consistency reasons. In other words, minimizing variation between runs in the handling of samples. Such automated workflows could be implemented on systems such as the Bravo or the Magnet if you would like to have a more walk-away solution. And then there's the data analysis, including secondary analysis such as sequence alignment and variant calling, as well as tertiary analysis with software solutions such as the ELISA interpret, which allows for clinical interpretation of the sequencing data. From this first SureSelect kit, it has evolved over time to what we have today is the latest DNA library prep solution in the form of the XT-HS2. This solution includes a range of features from molecular barcodes to the ability to accommodate various sample types, as well as having a more streamlined workflow amongst many other beneficial features. And in terms of target enrichment, as mentioned earlier, it could be from whole exome sequencing with the current iteration that we have in the form of the V7 design, which is implemented in a variety of different applications, from molecular characterization of rare diseases to tumor profiling for precision medicine. Or it can also be accommodated with not only just a whole exome design, but also other panels within the exome portfolio, such as the clinical research exome or the focus exome, as well as many other exome options. Now, should the Agilent catalog panels not able to meet the requirements of the researcher, the SureSelect platform has the flexibility to be able to allow the researcher, such as yourself, to customize a panel specific to your requirements in covering the regions that you are interested in for your research. The benefit for customization is manifold, including higher sample throughput, more cost-effective, so lowering the sequencing cost per sample, as well as faster time for sample to result. And the customization process is done through a web-based tool called SureDesign, where a very simple, user-friendly interface is available for you to create any panel of your interest. There is, of course, also another option with the design service, whereby the Agilent team is able to assist with the whole design process. Shown here is a publication example of a custom SureSelect panel. The researcher, in this case, is interested in developing a panel targeting 21 different genes for a breast and ovarian cancer panel. And in their testing of this design, not only were they able to detect the variants that is expected from the test population that they were working with, but they also had 0 false positive calls. And it is with this customization capability of SureSelect to be able to accommodate to the user's design requirements that the research community have explored and adapted it to other NGS applications, such as targeting non-human targets in clinical samples to enriching for ancient DNA genomes. There are also examples of targeting regions of animal and plant genomes, as well as cool as it sounds, insects. In addition, users have also explored into adapting SureSelect in epigenetic-related research applications. Now, high-throughput chromatin conformation capture, or HiChIP, is a technique used to generate genome-wide chromatin architecture maps. And while these maps are very informative, it can be limited in terms of its resolution. Now, when these maps are acquired with a higher resolution, unique details of individual chromatin loops or organization and dynamics can be observed. However, this higher resolution comes at a cost in the form of a significant increase in sequencing depth requirement per sample. And this can be as much as hundreds of millions of reads and even more. Hence, the whole genome HiChIP at a high resolution can make it cost prohibitive But without the higher resolution, it would lead to, of course, poor resolution and or exclusion of those valuable experimental conditions or controls. And to overcome this challenge, a targeted approach for HiC is explored as a potential solution. And as Anthony introduced earlier on, Agilent and Arima Genomics have partnered to provide a streamlined target enrichment solution for HiC, or otherwise known as Capture-HiC. This is done by combining the flexibility and customization capability of the SureSelect platform with the Arima HiC solution. This targeted approach allows the researcher to focus the HiC reads on the regions of the genome that is of interest, and this in turn reduces the sequencing read requirements, in other words, lowering the sequencing costs, with the overall goal of generating a high-resolution HiC map in those regions of interest. The overall Arima HiC SureSelect workflow is illustrated in the flowchart on the right, and it basically describes the workflow starting from the generation of the HiC library from the cross-linked cells. Then the HiC library is then fragmented prior to the preparation of the NGS library, which will then be enriched with the SureSelect XTHF target enrichment platform. These SureSelect probes or baits are customized to target regions of interest set by the researcher. And in the case of the AppNode example, 9 genes or gene clusters are targeted. Now, I won't go into further details in relation to the AppNode, and Anthony has provided a very detailed overview of the AppNode as well and some of the data. But I would like to just reiterate like what Anthony has just shown earlier. By doing the Capture-HiC, you're able to achieve a sub-kilobase resolution, as you can see in the figure at the bottom left-hand corner. And it is with this high resolution that enabled for a more precise identification of topological landmarks and finer scale observations, in this case, such as that of the chromatin loop anchored at the promoter intronic region of the BCR locus, as shown over here. Now, should anyone be interested in the AppNode, I'd like to reiterate like what Anthony has mentioned. Do let us know, and be more than happy to provide you with a copy of the application note so that you can take a look further into the details of the experiments. Now, other than Capture-HiC, another aspect of epigenetic research is DNA methylation. For DNA methylation, often it focuses on the CpG islands, and many of these CpG islands are found in the promoter regions. Hence, the methylation status of these CpG islands in the promoter regions have a direct impact or relation on the expression of the associated genes. This is of importance as it has been shown in many studies of the role of DNA methylation, for example, in cancer, such as hypomethylation in heterochromatin genomic instability, or hypomethylation in tumor suppressor genes. DNA methylation also has a role in other diseases such as neurodevelopmental disorders or imprinting disorders. Now, in a recent study, it was shown that not only is the CpG island of importance, but in the regions outside of the CpG island called CpG island shores and shells have shown differential methylation not only between different cell types, but also between the normal and tumor cells of the same cell type. So similar to the whole genome HiC, whole genome methyl sequencing or whole genome bisulfite sequencing can be cost prohibitive due to very high sequencing read requirements per sample. Hence, a targeted approach is also explored. The SureSelect human methyl seq design is an 84 MB capture, which covers not only the CpG islands, but also cancer and tissue-specific DMRs, promoter regions, and many other important targets. And while the methylation data may not be across the whole genome, it is still very comprehensive and is able to cover many of those important regions, covering about 3.7 million CpGs, which is more comprehensive than the methylation arrays. And this is highlighted in a publication example which had compared between the different technology options. And I found methyl capture to not only provide more comprehensive coverage compared to methylation arrays, but also showed strong correlation with whole genome bisulfite sequencing. In addition to the human methyl design or panel that was just mentioned, there are also examples where researchers have taken the customization route or approach for targeted methyl-seq, as shown here in this publication example, where a specific region was targeted to determine its methylation status and its length in prostate cancer. Now, apart from the catalog design that I just mentioned, there are also panels developed by researchers who have made their designs to be available to the rest of the research community via this Agilent community design platform. What this essentially means is that the Agilent community design are designs, whether it be NGS panels or even microarray designs or panels, as shown here as some examples that are created by our customers and has been deployed successfully in their laboratories. And for epigenetic research application, there are targeted methyl-seq designs for both the rat and mouse that has been designed by Dr. Richard Lee of the Johns Hopkins University. Now, the rat design is a 97 MB capture size, while the mouse is about 109 MB. And both of these designs are available and has been referenced in the number of publications as listed here on the right. Now, apart from looking at epigenetic changes, another aspect of research that may be of interest could be looking at the transcriptome itself, as well as the gene expression profiles. Instead of sequencing and analyzing the whole transcriptome, a more targeted approach could be implemented in order to focus on the targets of interest. And it is by focusing on these targets of interest that the overall approach is more cost-effective, even though you would be sequencing deeper in order to identify or be able to identify some of those rarer features, such as alternative splice variants, gene fusions, and of course, you still get the gene expression profile data. This is because, like Capture-HiC, the sequencing reads are focused to those regions that are of interest, and hence with the higher coverage allows for the detection even for those rarer features. An example of how targeted RNA sequencing has been implemented is highlighted in this publication from a group from Japan led by Dr. Mano. With the targeted approach, BCR-able gene fusion could be identified, and this was done even with only probes being designed to only one of the fusion partners. On top of that, targeting a number of other target genes, in fact, about 900 human kinase genes. And with the sequencing reads focused to those regions or targets of interest, Dr. Mano's team was able to achieve much higher coverage and hence able to identify more of those occurrences of features, such as alternative splice variants and gene fusions and so on. And this would not have been possible if they had taken the whole transcriptome sequencing route. Here, we also have another example similar to what I just shown earlier in relation to the work done by Dr. Mano, where again, gene fusion targets is of interest, in this case for the research related to gastric cancer. So like the DNA capture kit that I mentioned earlier, the RNA target enrichment solution has also evolved and improved over time to what we have currently, that is the XTHS2 RNA. And it includes features such as molecular barcodes, more streamlined workflow, as well as being able to accommodate low amount of total RNA input, as well as different type of total RNA, regardless of whether it be from FFPE samples or high quality total RNA. And while molecular barcodes may not be typically used for deduplication in gene expression analysis from the transcriptome data, recent studies have shown the benefits of deduplication using molecular barcodes can improve the expression profile reproducibility, especially for the low expressors. And as shown here, when we did some comparison from some of the internal data that we have made here at Agilent, as you can see, without deduplication, there is a systematic overestimation, right, compared to what is expected. And when doing the deduplication with chromosomal coordinates, there is an underestimation of the high expressors. But when we compare these data to deduplication by chromosomal coordinates with molecular barcodes, there is a greater correlation and hence a more accurate expression profile, even for low input and high sequencing depth libraries. And then finally, I'd like to very quickly finish things off and mention that apart from next generation sequencing, there are also other technologies that could be used for epigenetic studies, including microarrays in the form of methylation arrays or gene expression and microRNA expression microarrays. And when it comes to expression profiling, for instances where there are only a few targets of interest, qPCR technology can also be adopted to profile a small number of gene targets. And with that, thank you very much for your attention.
Perfect. Thanks, Jiang. Really appreciate it. We've got the agenda up here, and we've got just 2 minutes for questions. We check the Q&A, and I've got a couple of questions here myself. Okay. The first one is there any limitation to the size of custom panels for either DNA or RNA capture?
Yeah, so in terms of customization, there are officially, we can go up to 24 MB in size, but in terms of the technology itself, there really is no limit. As you saw for some of the examples earlier on, we have customers who have developed all the way up to above 100 MB. So technically, there is no limit.
Okay.
We just need to make more probes.
Yep, sure. No, fair enough. Maybe the last question is just due to time. If I'm interested in looking at methylation status in animal models that I'm using, not mouse or rat, would that be possible?
Yep. So in terms of the customization, so again, with the customization approach, it will be done through the Share Design platform, and there are a number of different species that we support within that user interface. So you can basically customize based on that. Now, if there are any species that is not supported, so there's some interesting ones or the ones which we were not able to access the genomes on, we can always get in touch with us, and we can always do a customization design through the design service. So that's also another option.
Okay, perfect. Thank you. Well, thanks, Jiang. Really appreciate the time and the content. Sounds like quite a few solutions from Agilent, which is great. And we're going to move on to some real research, which is fantastic. Just again, a reminder, Dr. Fulwood here has to leave at the end of her session. So if you have questions, please go into the Q&A box, hit all panelists, and please type your questions there so we can get them addressed during this session. Without further ado, our keynote speaker is Dr. Melissa Fullwood. Dr. Fullwood is an assistant professor in the School of Biological Sciences at Nanyang Technological University with a joint appointment as a principal investigator at the Cancer Science Institute of Singapore. She is also an adjunct principal investigator at the Institute of Molecular and Cellular Biology within A*STAR Singapore. Her lab works on understanding the roles of three-dimensional organization of our genome in transcription regulation in cancer cells. Melissa, thank you and welcome.
Yeah, hi everyone, and I'd like to thank Christian for the very nice introduction, and also thank you to Arima as well as Agilent for inviting me to speak today in this webinar. It's been very interesting to learn all about the technologies, and I've also worked a lot together with Arima for the past few years, and it's been really good to work together with both Agilent as well as Arima together on my lab project. So today I actually have two stories prepared. So story one is the story that I mentioned in my title, where we are investigating the three-dimensional organization of enhancers for something new. We are looking at silencers regulating gene transcription, and it's work done by Ph.D. student Zhang, who's not in this photo, as well as my postdoc HiChIP. It also involves another Ph.D. student who was the one HiChIP experiment that I mentioned at the end. And we also have story two, which is a very new work coming up from my lab. We have been optimizing. Oh, sorry, what's the issue with my audio?
On my side as well, it's just a little bit choppy.
Can people hear?
Maybe it's the Wi-Fi.
Oh, okay.
It's a little better now.
Okay, yeah, I think that it's a bit rainy this morning. Yeah, okay, great. Okay, so in the second story, which is a story about what we are calling biopsy, because we adapted the Arima HiChIP method in order to be able to study fine needle biopsy cancer samples, which are a very common tissue type, but not an easy tissue type to work with. That has been done by the postdoc Samhabi, as well as my PhD student Ruchi. So to start, I don't think I really need to introduce chromatin interaction too much. Basically, chromatin interaction is two or more regions of the genome that come together in close spatial proximity, and these include things like compartments, paths, and enhancer promoter loops, and these can be identified by the suite of C technologies, which range from 3C. I'm going to be presenting a lot of 4C data. So in 4C, what we do is that we will use investigate all the hits that match to a particular bait of our interest. So the previous work that I did as a PhD student was really to develop HiChIP, which is a sort of a precursor to HiChIP. But today I won't show any HiChIP or HiChIP data. I will be mostly showing IC data that we've done together with the Arima HiChIP kit. So to interpret the HiChIP heat maps that I'm going to be showing later on, what we've done is to identify some of the topically associated domains or tags, and these can be seen by looking at these images where you have one set of genomic coordinates and another set of genomic coordinates, then regions of contact enrichment. Regions of contact enrichment would be associated with these square-like structures, which would be taken to be tags. And if we were to rotate these square-like structures, we get this sort of rectangle. And what we can say is that these regions over here would be anchor 1 that loops over to anchor 2. And if we're going to show this entire process in 3D form, like a sort of a visual representation, this hair triangle could be represented as this sort of multiple kind of structure. So into super enhancers are regions that are in terms of the histone modification called H3K27 acetylation. And so these regions have got multiple enhancers that are very close together, and we think that collectively they may be able to very strongly drive chromatin, to very strongly drive gene expression. And in previous research, my lab also found that these super enhancers are more associated with chromatin interactions. So then in our first story, we wanted to explore, are there looping silencers in the human genome? So we knew that regions marked by H3K27 acetylation, H3K27ME3 are known to harbor silencers. However, there were actually very few silencers examples that were known in humans. There have been efforts to look at silencers that were done in mice, as well as in Drosophila, but there have been very few silencers known in humans, and there have been almost no distal looping silencers that have been characterized in humans thus far. So what we hypothesized was that similar to super enhancers, can we find super silencers using a similar approach? Can we also try to look for peaks in the H3K27M3 signal, and then to them, and then to get signal ranking, and then to call these high regions as our MRRs, or that stands for H3K27ME3 rich regions. So we hypothesized that these can be a rich source of silencers, and maybe they can function to affect gene expression by both in terms of proximity as well as via looping. And so we did this analysis, and then we associated our MRRs together with HiChIP data that was published by the Agilent lab. What we found within K562 cells, what we found was that the constituent peaks, MRRs, tend to have more chromatin interactions as compared with typical H3K27ME3. And then we looked at genes that were associated with MRRs, and we found that the genes that are both associated with MRRs by proximity as well as distally, they tend to have gene lower expression as compared with other control genes that are not associated in any way with MRRs. So next, we wanted to characterize these MRRs by using CRISPR excision, and so this is showing MRR1, which is a region that we called from this H3K27ME3 data, and you can see here that there is this is the HiChIP data. So over here there is this anchor that is at the FGF18 gene, and this will loop to this anchor that we call A1, and this loops over to this anchor that we call A2. And so over here, what we did was that we CRISPR excised out anchor A1 after having verified by 4C that indeed it does have chromatin interactions to FGF18, as well as it turns out that we found that it also loops to other genes, thus suggesting that maybe this anchor A1 could maybe regulate multiple genes, not just one gene. And so we performed RNA-seq after having obtained CRISPR knockouts, where we excised this 1 kb region with the anchor A1. So what we found was that this FGF18 gene as well as this UBT2 gene is upregulated, and when we did, so in order to confirm this, we performed RT-qPCR, and so we found that indeed FGF18 was significantly upregulated, as was UBTB2 and FBXW, which is a gene in which A1 was associated with this intron, that did not have any change in terms of gene expression. And in order to address issues of heterogeneity of the clones, where we also generated 2 additional knockout clones, and by looking at this by RT-qPCR, we could also see that FGF18 was again upregulated. So next we performed CRISPR excision of MRRA1. Next we investigated this CRISPR excision results, and what we did was that we did a gene ontology analysis, and we found that there were some terms that came up, such as cell adhesion, migration, proliferation, differentiation. So then we investigated whether our knockouts have alterations in terms of the cell differentiation by looking to see whether there were hemoglobin genes that were associated with the erythroid differentiation pathway. And we found that compared with the empty vector, these genes started to be more highly upregulated. At the same time, we also found that the knockout clones tended to aggregate together into clumps, which we could also quantify using a cell adhesion assay, thus indicating that there were indeed changes in terms of the cell phenotype. Additionally, we performed 4C at the FGF18 gene and promoter, and we found that there were alterations in terms of chromatin interactions. In fact, we found that there was a gain of chromatin interactions with this other anchor, A2, and we also found that there were gain of chromatin interactions outside of this region over here. So we also wanted to know whether or not another region would also show this gene upregulation. And so what we did here was that we chose this other example, MRR2, and this has got chromatin interactions between the regions A1, A2, and A3 that all go to this IGF2 gene. And using 4C, this IGF, we can see that this IGF2 gene is indeed interacting by chromatin interactions. Next, what we found was that by doing RT-qPCR on 3 knockout clones, we were indeed able to see upregulation of the IGF2 gene in these knockout clones. Thus, this indicates that both MRRA1 as well as MRR2A1 can function as silencers of gene expression as determined by CRISPR testing. So next, we collaborated with Professor Vinita as well as Dr. Mani in IMC, and what they found was that these MRRA1 knockout and MRR2A1 knockout, they actually show reduced tumor growth within mouse models, within xenograft mouse models. We can see here that there was reduced tumor volume, and also some of these cancers just did not grow within mice. And you can also see for this MRRA1 knockout that all of these cancers look significantly smaller as compared to our negative control. So the next question that we asked was, how do the silencers at the two chromatin interaction regions anchors function together? Could it be additive or could it be synergistic? This is one of the questions that has been actively being debated in the case of super enhancers. And now that we have described a new phenomenon of super silencers, we wanted to dissect the functioning. So here we use the FGF18 gene, and we investigated what happens when we got A1 as well as A together. We would expect to see a much bigger consequence if only if we were to do a double knot. And indeed, that's what we found. This is the qPCR of FGF18 in the A1 single knockout. When we knock A2, we again see that by RT-qPCR that FGF18 is upregulated. And in terms of the double knockout, you can see that the axes are now different over here. Looking at the double knockout, we actually found a much greater increase as compared with the empty vector as compared with the A1 and the A2 single knockout, thus suggesting that FGF18 is upregulated synergistically. We were also able to see this by RNA-seq. next, we obtain growth curves in vitro of our knockouts. So this is the growth curve for the A1 knockout, and this is the growth curve for the A2 knockout. You can see here that as compared with A1 and A2, the double knockout growth curve is much more significantly downregulated as compared to the A1 and A2, thus indicating that there is much greater cell death. And we also performed a caspase activity assay because we thought that this might be associated with greater apoptosis. And indeed, you can see that the double knockout has got much greater caspase activity as compared with the empty vector and the A1 and A2 knockouts. And because we also again found that this was associated with cell differentiation, so we performed the RT-qPCR of the hemoglobin genes to investigate whether there might be increased erythroid differentiation. And what we found was again that the double knockouts show much greater increases in terms of the hemoglobin gene upregulation. And also, we again collaborated with our collaborators at IMCB, and we found over here that in terms of the double knockout xenograph, there is much more significant tumor growth inhibition as compared with our single knockouts. So next, we asked, as I mentioned earlier on, we found that A1 knockout has got increased interactions with to the A2 region. So we were wondering if there perhaps some compensation by A1 for loss of by A2 for loss of A1. So maybe it's the case that you have two people working in the lab, and now one person is off sick, and so maybe person A2 now works harder to try to make up for this loss. But if you have both A2 and A1, then there's nobody working in the lab anymore, and of course the lab cannot function without anyone working in it. So now we have A2. So now that we have A1 knocked out, and then in our double knockout, we had A2 knockout. What we saw is that actually we see a great loss in terms of chromatin interactions. Thus, we think perhaps this loss of interactions might perhaps underlie this synergism that we see. So next, we asked whether EZH2 inhibition might lead to any upregulation of MRR2 associated genes. The reason why we looked at EZH2 is because it's a component of the polycomb repressor complex that will deposit H3K27Me3, and if we inhibit EZH2, that leads to reduced H3K27Me3 levels genome-wide. So we first looked at the RNA results upon performing EZH2 inhibition, and what we found was that the genes that were associated with MRR were most upregulated, followed by the typical H3K27Me3 genes, and these were both higher as compared with genes that were associated with super enhancers or typical enhancers. And performing at FGF8 motor, we found that there were chromatin interaction changes, and it tended to be the more long-range chromatin interactions that were either gained or lost. And also, we did this in our knockout cells, and what we found was that this attenuation of this upregulation of upon EZH2 inhibition, which leads to upregulation of IGF2 and FGF18, this was partially abrogated by having knockouts. The knockouts, as you might expect, if it were the case that this silencer A1 and A2 was indeed regulating these target genes. So finally, one of the experiments that we did together using the Arima HiChIP kit was that we performed HiChIP of EZH2 inhibited as compared with DMSO treated cells, and we found that there were many head changes in terms of our HiChIP data, and we found that at FGF18 there was this compared over here in the MSO, this region does not have a tag called, so it was not considered significant enough to call a tag. However, in the EZH2 Rep1 as well as our Rep2, we found that this region had called. And this, when we overlay this data together with our 4C data, we can see that this tag actually very nicely appears between A2 and A1. So we're just wondering whether perhaps these tags might to explain the localization of these chromatin interactions in these specific locations. So in terms of conclusions, in terms of our story one, we found that there are indeed changing chromatin that there are indeed that the knockout indeed leads to increase in terms of the gene expression of some of our genes, and in terms of the chromatin interactions, these changing chromatin interactions might underlie the synergistic functioning at the FGF18 looping silencer locus. So our next story is a more clinical story, and what we wanted to do here was that we were hoping that in the future HiChIP might be able to have clinical applications. So there has been great interest in terms of performing HiChIP clinical samples. This has been done for some liquid cancers, the TAL experiment that's from Ioannis Ifantis' lab, which Anthony presented on earlier. And we have also done some experiments together with acute myeloid leukemia and chronic lymphocytic leukemia. But these have been mostly possible for liquid cancers, for which it's possible to get a lot of to get sufficient cells, and also some solid cancers, for which it's also possible to get quite a high number of cells. So we really wanted to be able to generate high-quality genome-wide chromatin confirmation maps from needle biopsy samples, which you can see over here are very small. So one of the first challenges is in terms of the cell number, but this cell number is not that small. The cell number is about 100K cells. So, however, so in terms of the cell number, that is not the most major impediment to working with these samples. The major impediment is that these are tissue samples, and thus we needed to be able to have some way of preparing these tissues. We actually did indeed try to just use the standard HiChIP protocol without doing making so many changes, and this actually failed the first time round. So what we did here in order to try to improve things was that we adapted the in situ HiChIP method into what we call biopsy. So what we have here is a frozen tissue sample, and then we pulverize the tissue using a liquid nitrogen cooled tissue homogenizer. It's a mini mortar and pestle, which will be able to keep the biopsy sample frozen, and it will remove any degradation of the sample. And then because Arima is one microcentrifuge tube protocol, so this same microcentrifuge can then seamlessly connect with the rest of the Arima HiChIP approach. And so that's what we did. After homogenizing, we simply performed the rest of the Arima HiChIP experiment and obtained our HiChIP data. So we performed this using 3 nasopharyngeal cancers, which is a cancer that is quite prevalent in Southeast Asia as well as the rest of Asia. It's basically a nose and pharynx cancer, so it can either present on the nose or in somewhere a bit deeper within the body. And so we obtained 3 of these fine needle biopsies from our collaborator, Dr. Go Buncher over in NUH and also at CSI. And with these 3 samples, we were able to get high quality genome-wide chromosome confirmation maps, S9, S10, and S12. We also did regular HiC on HK1, which is a nasopharyngeal cancer cell line. So this is the summary of the HiC data that we obtained. And then we were able to call tags as well as loops using Juicer, and also because fires have been a novel class of chromatin interaction types that were described by the Bingren lab. So we also performed fire calling, and we were also again able to identify these fires. Also looking at these maps, we were also able to see chromosome compartments as well as translocations, such as this translocation over here. So next, we investigated our HiC heat maps at these important oncogenes such as RUNX1, MYC, and EGFR, and we were able to see these large tags as well as some specific loops, for example, these loops over here and over here. We were able to detect these loops at the MYC locus, connecting together this long non-coding RNA CC81 as well as MYC, and they also connect together super enhancers together with these important oncogenes, and also because a fire, which mentioned over here, was introduced by Bingren and colleagues in I think this is actually Anthony's paper, Schmidt et al. So fires are local interaction hotspots, which show unusually high levels of local chromatin interactions, and they're these very tiny square-like structures over here, and we were able to see this type of fire at this EGR locus. And although I don't show the results here using the algorithm published in this paper, it was indeed called as a fire. And here are just some additional examples of oncogenes as well as long non-coding RNAs that can be regulated by these fires in NPC. And they're also associated with super enhancers that can be detected within these NPC samples. So next, we analyzed these fires from our data, and we found that first of all, we can see that there are a lot of fires, and 2, that they are associated with target genes via target genes. So in terms of the conclusions for our story, first we developed a biopsy method that can be used to generate high quality genome-wide chromosome confirmation maps from this new category of needle biopsy cancer samples. And because these tiny needle biopsies are actually used very much for the diagnosis of several cancers, and you really don't want to have to do more procedures on the cancer patient who's already suffering from cancer and does not really need to see the doctor even more. We think that this is a good approach to being able to do some, perhaps in the future, as a diagnostic and prognostic tool for needle biopsies of solid cancer in the clinic. For example, the detection of fires at key oncogenes that can be therapeutically targeted, perhaps identification of translocation, because over here we saw that our biopsy samples could indeed demonstrate detection of translocation as well as various confirmation features that we tend to see in the chromatin interaction landscape. So finally, with this, I want to thank HiChIP and who, as well as Ethan, who were responsible for story 1, as well as Sam Harvey and Ruchi, who have been pioneering the work shown in story 2. And with that, thank you very much for listening to this presentation. Thank you.
Thank you, Melissa. I appreciate your time and love the application name biopsy. Perfect. We have a question here in the Q&A box, and I'll read it for those that aren't on their computers. Is silencer promoter interaction insulated by tag boundaries?
Yes, so I think that it is insulated by paired boundaries. So if you see this, when we overlaid this result over here, you can see that this, that's the,
we might have taken the ball away from you there.
Oh, I see.
There we go, it's back. You can share your screen.
Oh, okay, great. Yeah, so if I just go over here to this slide over here, these were the paired boundaries that we called from K562 cells treated with DMSO, and you can see that this actually very nicely follows that our 4C identified chromatin interactions fit quite nicely within these pairs as well as these subpairs. So I think that there is some relationship between paired boundaries and silencer promoter interactions. Yes.
Okay, great. Sorry, you weren't sharing your screen there, but I think the explanation suffices.
Okay.
Did you observe, or do you observe these interactions primarily in the B compartment?
Yes, that's a very good question. We have not actually done a lot of analyses of the B compartment, but I would imagine that that is the case, but we just haven't done that analysis yet.
Okay. Okay, just double checking the Q&A box, and I think that is all. We are actually a couple minutes over, so we'll have to wrap it there. Thank you again, Melissa, for the time. We really appreciate it.
Thank you.
We're on to our last session of the day. Thanks for everyone for sticking with us. Hopefully it's been informative morning for you all. We are joined by Dr. George Nene. George is an assistant professor and research fellow at the Cardiovascular Research Institute at the School of Medicine at the National University of Singapore. George is a clinician by training and has worked at the front line of Nigerian hospitals, treating patients with resources that are vastly more limited than what we are accustomed to in Singapore, and I'm sure as well here in the States. He has now exchanged his stethoscope, for a pipette, qualifying with the National University of Singapore Ph.D. in cardiac stem cell bioengineering within Henry Yu's lab. George is now making further inroads into cardiovascular epigenetics and epigenomics in Professor Roger Fu's lab. His projects look at DNA damage and cardiac genome enhancer promoter interactions. George, thank you for joining us. The floor is yours.
Hi, good morning all. Can you hear me?
We can, thank you.
Yes, all right. Good day all. So today I'll be sharing some of the work we're doing in our lab, in Roger Fu's lab, and looking at enhancers and understanding the importance of these enhancers in cardiovascular disease, particularly looking at how mutations, the variants that fall on these enhancers could regulate disease and also could lead to identifying novel targets for heart failure. Right. So, so we know that heart failure is the leading cause of death and morbidity worldwide. This is true in both developing countries and even in developed countries. And it's particularly important for Singapore, where about 5% of people are living with heart failure. And this poses a huge burden to the healthcare system in Singapore and around the world. Now, heart failure is often characterized by some classical gene expression changes. As you can see in this slide, as it goes from a healthy heart to a compensated hypertrophy and to a decompensated hypertrophy, we find a classical expression of genes. Fetal genes, for example, get reactivated in the heart failure, while you have a downregulation of genes involved in calcium handling. And this gene expression changes even get worse as it goes for decompensated hypertrophy, and leading to a dysregulated pathways that then lead to the heart failure phenotype we see. And so we believe that regardless of the underlying pathology that gives rise to your heart failure, be it due to your lifestyle change, genetics, or things like hypertension, it often acts to modulate the epigenome and give rise to this because regardless of what the underlying cause is, you have the same gene expression changes. And so this then leads to the various pathways that are altered and gives rise to the heart failure phenotype. And so like I mentioned earlier, we're looking at two main important aspects of the non-coding genome or the epigenome. And the first one is the enhancers, and looking at the importance of enhancers in regulating the gene expression changes which you have in heart failure. So in the previous videos I've talked about regulatory regions, and I want to again recapitulate what I said earlier. So enhancers are known as this the DNA sequence non-coding, often marked by H3, K27AC marks and K4ME1 marks. They're usually bound by transcription factors, and they engage in long-range enhancer-promoter interactions. And thanks to this enhancer-promoter interactions, it regulates their target gene expression. And now the other aspect of the non-coding genome that we're interested in is looking at 3D architecture, and this is where the Arima HiChIP comes in very handy. Again, just to recapitulate, basically what you do in your HiChIP experiment is that you cross-link your chromatin, your DNA, you cut with certain restriction enzymes, and then you fill the ends with biotin. And the idea is that when you religate, the regions that are close in proximity get religated to each other. And then when you share and pull down your using a streptavidin base with the biotin, and you sequence, you understand, you unravel the various regions in the genome that are in contact with each other. And so in a lab, we've done 3C, 4C, and HiC and HiChIP, again, using the Arima kit. And thanks to this experiment, we understand that the genome is compartmentalized. So you have the inactive compartment often at the periphery of the nucleus, and the active compartment often at the center of the nucleus. This chromatin. And then when you zoom in deeper, you understand that it has these things called TADs, topological associated domains. These are regions of high interacting frequency within which you often have this transcriptional regulation between the enhancers, regulatory elements, and the target genes. And they're often bound by architectural proteins such as CDCF and toficin at the boundaries. And so it gives you a map like this, which again we've seen in previous slide, where you have this triangle, which are the TADs, and this red dots indicate regions of high interacting frequency. And then you have this strong dots at the apex, which indicate that these two regions form a tight loop. And as you can see that at the boundaries, they are often marked by CDCF based on our chip sequencing here. So now, using these two tools of the enhancer chip sequencing and 3D architecture, we went in to look at the various SNPs that fall on enhancers and see how we can use that to understand or identify novel targets for heart failure. And why is this important? This is because majority of all the genome-wide association studies, the disease-relevant SNPs often lie in non-coding regions. As you can see clearly here, only about 5% fall on coding regions. A majority fall on the non-coding regions. And of this non-coding SNPs, most of them fall in regulatory regions marked by DHS, DNS sequencing. These are active open chromatin regions. And so it often makes it difficult to assign biological importance to these non-coding SNPs because as they don't fall on coding regions, it's often difficult to understand what they are doing. And so to answer this question, one of the approaches we took was to look at chromatin QTLs. And what are chromatin QTLs? These are SNP mutations that correlate with changes in the chromatin state. In our case, we look at something called HiChIP, histone acetylation quantitative trait loci. And for this, we used a tool called the GSI test, which was developed by a researcher in our institute, Prof. Shyam Prabhakar. Basically, what it does is that it detects and correlates SNPs with chromatin state variation. In this case, we're looking at changes in peak height. So in this example here, you see that when an individual that has a G and one that has a C in the same location, the same position, is often associated with a decreasing peak height in your chip sequencing. And when you have a HEC, it's often somewhere in between the G and C. And so what we did was we performed enhanced chip sequencing on 70 human hearts. And we have about half of them are non-failing hearts, and half of them were failing hearts, human left ventricles only. And then we performed the chip sequencing, enhanced chip sequencing. We performed RNA sequencing, HiC and HiChIP using Arima kit. First, we studied the various changes in the acetylome, comparing the healthy to the diseased heart, to understand the regions that are changing. And then next, we applied the GSI test to look at the various SNPs that correlate to changes in acetylation peak height. And then we overlapped our study with relevant GWAS studies to understand if we can identify novel targets of these SNPs. Right. So to take you through our data, first we looked at the changes in the histone peak between the heart failure and the non-failing hearts. And as you can see here, we identified close to 4,000 differentially acetylated regions, about half. More were going down than the ones were going up. And so using the TF motif discovery tool, we identified important transcription factors which were involved in regulating the changes in the heart failure. And I want to highlight key TFs, such as the MEF2 protein, which is known to be important for heart development. And as you can see, we identified ATF3 as well. These are known and important transcription factors that are involved in heart regulation. Performed the great ontology analysis, and we showed that indeed many of these changing regions were involved in pathways related to things like cardiac differentiation, and as well as inflammatory response. These are pathways that have been shown to be triggered in heart failure. So next, to show the correlation between our chip sequencing and our RNA sequencing, we try to correlate the various changes in histone peak height with the RNA sequencing. First, we used distance cutoff, and we show that indeed, within distance, we see a high correlation between when the RNA epigenome is going up, the fold change, you have a corresponding increase in increased peak height. But to further to show the importance of using the 3D architecture, we show here that by using also our chip, HiChIP, you see a nice correlation between when the RNA sequencing is going, when the RNA is going up, expression is going up, you have a concomitant increase in the acetylation peak height. And as shown example, I want to highlight this. Here we see an example of a loci, a distal enhancer, which is low in non-failing hearts but increases in heart failure. And our HiChIP experiment linked it to this gene, BMP4, located about 100 kilobases away. And using, by analyzing both the RNA sequencing and the chip sequencing, we see that indeed, when the acetylation marks are going up, the gene expression is going up as well in the heart failure. Right. So now that we've analyzed and seen the changes in human heart failure compared to the non-failing hearts, next we went on to look at the SNPs that we identified from our chip sequencing in the enhancer regions. So applying the JTK standard tool, we identified close to 250,000 of SNPs that fall into enhancer regions. Most of them have been previously identified in genome match, only about 0.5% we are about. We validated this SNPs using Sanger sequencing and about 10 of these loci, and we did show that indeed these are accurate. We do see the change in the heart that were identified to harbor the SNPs. Then next, applying this GSI test tool, what we were able to do was to look at the SNPs that correlate to changes in acetylation peak height, and we identified about 1,680 HiChIPs. These again are mutations, variants that correlate significantly to changes in acetylation peak height. And I show here an example. We have the ones that are going up to show an example. This is a SNP located in the intron of KLX12. You see individuals that have an AA versus some have a GG, an increase in peak height. And this increase is associated with an increase as well in KLX12 expression. Similarly, examples of ones that have a decreased expression, you see this enhancer, individuals that have a T as compared to ones that have a G have a reduced peak height. And as you can see as well, the reduced peak height correlated with reduced expression of the gene. So next, using again our HiChIP experiment, we're able to identify target genes of some of this HiChIPs. And I show you an example. This NOS1AP. This is a pig or SNP, which was associated with an increase, as you can see here, the change from a C to a T with an increase in peak height. And we show that this is associated with a nearby gene, NOS1AP, which is next door, but it doesn't have any effect on the OLFM2B, which is quite close to it. And this is because it is separated by a CAD boundary, making it therefore isolated from this enhanced activity. But interestingly, using our HiChIP, we also identified that this SNP also was associated with a very distal gene, C1RF226, which, as you can see here, it was linked using it has a GTEX EKTL, and also our HiChIP was able to link this SNP to this gene, showing the importance again of looking at 3D architecture in understanding the target genes of these enhancers. And so finally, we try to understand why these SNPs could affect enhanced activity. And so we turned our attention to look at the TF motifs. And as you can see here, indeed, we see that interestingly, when a SNP or HiChIP led to a gain of some non-cardiac relevant transcription factors like the MEF2 family, it was often associated with an increase in peak height. And when you lost the MEF2A, it was correlated with a decrease in peak height. Similarly, we had some repressive transcription factors like the ZEP1ID4. And when you gained this motif, you were more likely associated with the decreased peak height, and when you lost it, you had an increased peak height. And then overlapping our 1680 HiChIPs with GWAS studies, we were able to identify 62 potentially new targets, novel targets, that could be relevant for cardiac disease. And I want to highlight that already 2 of such have been published recently by different groups. One is the MK5 and the other is the MTSS1. These studies basically validate our finding that using enhancer SNPs, we could identify potential novel targets that could be important in heart failure. And so for this first part of the study, basically we've performed, we're the first to perform large-scale, large-scale human heart enhancer and 70 human hearts, identifying about 47,000 enhancers. And then by applying the GSI test, we've been able to identify 1680 HiChIPs. And we showed that this correlated gene expression by using architecture 3D enhanced experiments. This study was published recently in Circulation Research, and it was on the front page. Now, on the last few slides, I want to show a second work we did, also looking at enhancers and trying to identify top regulatory cardiac enhancers, because we believe that by identifying this, again, just like we looked at the SNPs, for example, rare mutants in these enhancers or deletions in these enhancers could help unravel the molecular mechanisms of some mutations and how they're linked to disease. So for this experiment, we looked at this algorithm called the ABC, activity by contact. So some of background, basically, in trying to identify enhancers, there are often a number of approaches that one could take. You could look at chip sequencing or HiChIP sequencing or DNA sequencing. These assays often identify the chromatin, open chromatin regions, the regulatory regions, but they don't tell you the target gene. On the other hand, assays like the HiC and HiChIP and Capture-HiC could help you link putative regulatory regions to their target genes, but they often don't tell you how strong a regulatory impact that enhancer has on the gene. And so this algorithm called the ABC, what they've done is that by integrating three key experiments, your chip sequencing, your DNA, so HiChIP, and your HiC, and by applying an algorithm, you're able to score the enhancers from 0 to 1. The higher the score, the higher the regulatory impact of the enhancer on the gene. The lower the score, the lower the regulatory impact. And so, to validate their study, they performed a CRISPR-I experiment in their cell type of interest, and they showed that roughly using a cutoff of 0.05 of this ABC score, you find that many of the genes that score higher than 0.05 are more likely to have a direct impact, at least at baseline, on the gene expression of the target gene, while enhancers that have a lower score often have little to no effect of the genes. I must mention that enhancers could act at baseline, or at times you need to have a stimulus for to see an enhancer play effect. Regardless, it's a good algorithm, it's a good tool to help you, at least in every context, to understand what are the top regulatory enhancers for the genes of interest. And so what we've done is that we applied this algorithm to our human hearts, human left ventricles, from the chip sequencing, HiChIP sequencing, and HiChIP using the Arima kit as well here. And for in vitro validation, we performed the same in our embryonic stem cell derived cardiomyocyte to generate the ABC scores for both the human heart and the in vitro model of HiChIP cardiomyocyte. And we saw that indeed the ABC score was quite similar in both the human heart and in the HiChIP cardiomyocyte, with an R value of about 0.87, showing that indeed we could use our human embryonic stem cell cardiomyocyte model to validate the enhancers that we've identified from our human heart. To show an example of what this looks like, this is the gene, a cardiac gene, ACTC1, is the known character-specific gene. And so you see, if you use a regular Capture-HiC or HiC, it could identify these three targets, labeled as E1, E2, and E3. But it doesn't tell you which of these enhancers has a higher impact on the gene. With the ABC score, you now can tell that this one, located further away on the E2 and keeping a nearby gene JD2, is the top ranking enhancer for the ACTC1, while this is the number 2 and this is the third enhancer. And so, to actually kind of revalidate or show the utility of this ABC algorithm and identifying enhancers, we compared this with two other methods, Capture-HiC and HiChIP looked. And so to do this, what we basically did was we went to the GTEx portal and downloaded all the EQTLs. EQTLs meaning enhanced expression quantitative trait loci. And we looked at the ones that fell on enhancers, cardiac enhancers determined by the left ventricle, ENCODE left ventricle, H3, KIT, and 7AC. And they must be located at least 5 kilometers away from the nearest gene. So there are 340 of such enhancer EQTLs. And so we compared how well performed the Capture-HiC or HiChIP and ABC in predicting the target gene. We used a study was published in Capture-HiC in the human heart. And using the Capture-HiC, we were able to predict about 22% accurately. I must mention that this study actually gives the very same analysis on in their paper, in their published paper, and with similar results. This was higher than the random, obviously. It was significantly higher than random, but as you can see, it wasn't very accurate. The HiChIP looks seem to do better with the HiChIP. We did enhance the HiChIP, so we're basically focusing on only H3, H7, AC. I must mention that the Capture-HiC was looking at all promoters, they expressed or not expressed in the heart. It was just looking at all promoters and looked at all the possible interactions with both enhancers and non-enhancers. But for this study, was looking at enhancers only. Now, using the ABC cutoff of 0.05, we saw that more than half of these were eliminated from the list. That is to say, many of these enhancers didn't meet the 0.05 criteria. They scored less than that. But of the 122 that scored above 0.05, it identified 71%, about 782 else, showing indeed that the ABC score at least helps you to prioritize the top ranking enhancers, the enhancers that are more likely to have a direct impact. Because as we know, oftentimes one gene may have interactions with more than one enhancer. So the ABC more or less lets you know which of them has the stronger impact on the gene of interest. And so for further validate the ABC, we identified 3 enhancers for some cardiac specific genes. One of them is the ACTC1, which I alluded to earlier, the MYL2, and the MTSS1. And we showed that by deleting these enhancers, we're able to get between 20 to 80% reduction in expression of these genes. And so it was also published as well in circulation research with the paper. And so finally, summary, the activity by cluster algorithm helps identify key enhancers for cardiac genes, outperforms other models, and we showed by deleting these enhancers, you will be able to get a regulation of target genes. And to end, I want to say thank you to my mentor, Prof. Fu, and to all the members of the lab from various hosting, is a mathematician who has helped all analysis of the HiC experimental and chip sequencing data. Mick was very helpful with cell culture, and Eleanor helped with the HiQTL study. Thank you very much.
Thank you, George. I like the content, comparing and contrasting and bringing together a few different technologies. I've got a couple of questions here that came through in the chat, and then again, just a chance for anyone else to add some questions to the Q&A box. First one is, given that there are different cell types in the heart, are you able to tell in which cell types your SNPs are more active? Oh, we might have lost you.
Yes, sorry, I had the mute. Yes, so thank you for the question. Yeah, that's interesting. So the left ventricle contains, as you rightly say, different cell types on the heart. So what we've done in the next study is that we're trying to tease apart these various cell types by doing chip sequencing and HiChIP and ATAC on all the various cell types of the heart. We want to see if we can identify in which cell types these SNPs are more relevant. And so while we cannot tell accurately using our whole left ventricle because it contains all cell types, the next step we're taking now is to look at this and to identify the various cell type specificity. Thank you.
Okay, great. The second is, have you performed any chip experiments to confirm the predicted gain or loss of the TF motifs? I've lost you again.
Yes,
sorry. No worries. I do all that.
Yes. So we have done that as well, and we also did the gel shift assay, EMSA assay, to identify the SNPs. I didn't show the result here, but yeah, we've been able to show that, for example, the ones that have the MEF2 loss, indeed using our EMSA assay, we're able to show that indeed the disruption of this, the gain or loss of this SNP led to a loss of the binding of MEF2. Thanks.
Okay, great. I'm just checking the chat and the Q&A. I don't see any more questions. I just wanted to say thank you for your time, George. Great story, and thanks for wrapping us up today. With that, we have actually through the top of the hour for Q&A. I know there's a question quite a while ago for Anthony that we didn't get to at the start. And while we have you guys all thinking about Q&A, we're also going to throw up a couple of questions in a poll that will show up on the right side of your screen as well. We would appreciate 30 seconds of your time just to provide some feedback on what you thought about the schedule today, the content, how well you thought the host did, this guy, and all of that. So please send your questions through the Q&A, and we'll wait just maybe 30 seconds or so for your answers in the poll. We appreciate your time. Okay, I will do one call for questions. I don't see any popping up in the Q&A. I'll scroll back to the question I saw in the chat quite a while back. I'm not sure. I think Anthony might still be on. It's getting late here in San Diego. Anthony, do you happen to still be with us? Perfect. We had a follow-up question, I think, to the question that you received. It says, so it's quite a while ago, so kind of go back a few hours. It says, further regarding the HiChIP, do you perform, do you see that question there?
I think I read it when I came in about the IgG.
Yep, exactly.
Yeah. We haven't been doing IgG controls. I guess in the case of HiChIP, the background is HiC, whereas in ChIP-seq, I guess the background is more like genomic DNA. We do a couple other things to check for the enrichment. I think I showed that there was like a metaplot analysis that we did where we pile up our HiChIP reads around known ChIP peaks if you have known ChIP peaks in that sample. And we can calculate the peak height relative to the local background. It's nice to know what the background level of enrichment is if you just sequence a HiC library and you align that over the ChIP peaks. Usually there's some small level of enrichment. So that's one way of thinking about, I guess, how to compare your level of enrichment to background. And then there's another method out there called aqua HiChIP, which I think tries to achieve some type of cross-sample normalization with an interspecies spike-in. We have some customers who are doing that, and that's something else that we've been thinking about implementing going forward as well.
Okay, great, thanks.
Sure.
I don't see, let me see here, no other questions. I'll pause for just another 5 or 10 seconds, allow you to finish your poll, think of any other questions before we wrap it up. While wrapping up, maybe I'll just say thank you to our speakers. Really appreciate your insights from Melissa to Chen to George. Great content. Anthony and Chi Yang wanted to say thank you to you gentlemen as well. And thanks to all the attendees for sticking around with us for a few hours in your morning over there in Asia Pacific. Getting late here in California. Thanks again for your time and attendance. We really appreciate it. Any time away from the bench is very important. So thanks for spending it with us. We'd love your feedback again on the poll. And wanted to say a quick little shout out to my colleagues from Arima and our colleagues and friends out there in APAC on the Agilent side, Pearl, Charmaine, great working with you. With that, I think I don't see any more questions. We will wrap it up and close the day. Cheers all. Have a nice evening.